Great. Tell me when. No.
So just let me introduce a friend. I cannot say old friend because
Yeah.
very young, but
Oh.
we
Older.
were together several years, and he comes. If you want to see a real scientist, that's an intellectual. That's Joe, and it's fantastic to exchange ideas with him. He has immediate understanding and recall, and total recall, and it's wonderful to talk to him. He was really privileged that in his busy, big, super busy schedule, he got time to come here, and everything that he does and says is cutting edge and very fascinating. And maybe things that don't belong to the future, but to the present, but to the future. So please.
Thank you so much. Thank you. It's great to be back. Thank you, Rabbi Rappaport, and a particular thanks to my two daughters, and my daughter and my daughter-in-law, who are both students at Bar-Ilan, and I wasn't sure how much of an audience we'd have, so I wanted to make sure that. Thank you. Pack the audience. Right. I'm sure they'll have some good questions too. Anyways, thank you both for coming. Great. So what I wanted to do today, and it's wonderful to be back, is to talk a little bit about some advances both on the silicon side and on the neuroscience side, and how those two areas are coming together and some of the implications those might have. Want me to repeat? No. Some of the implications those might have for ethics and some of the moral issues that we think about, both from a religious standpoint, but also from a practical standpoint, from a technology standpoint. As you know, many of the technologies that we are beginning to roll out that involve AI and artificial intelligence, how do we dial in the right set of ethics into those new technologies that are rolling out? So let me start in the beginning. These are two pictures. These are taken both under the electron microscope, which is by now an old tool, but still an incredible tool for seeing things below the wavelength of light. So light has a wavelength on the order of a micron. If you want to see things that are well below that, you turn to a microscope that uses particles that have a wavelength that is much smaller, on the order of nanometers or so. And this is what a typical microcircuit looks like. This is all of the devices and things that we use are built principally on silicon, things that emit light. We'll take that off for now. It's fine. Everything that processes logic in general is based on silicon. We have other semiconductors, things like gallium arsenide that can emit light, things like OLED screens or LEDs. But all of the logic devices that we build are built on silicon. They're built by a process called photolithography. Photolithography is great because it gets better and better geometrically, doubles the number of transistors per unit cost per dollar every 18 months. That's called Moore's law. And so reliably we can predict how much compute processing we will have in 10 years from now, and it's a large number. And I'll show you some examples towards the end of this talk. A typical state-of-the-art microprocessor might have on the order of 50 billion transistors in it. It has a topology which is known as Manhattan topology. It's laid out in rows and columns. There's actually several layers to that. It's about 10 layers of metal connection layers, about 10 layers of connection inside of a typical chip. And that gives a typical connectivity which is on the order of 7 connections per transistor. So you've got 50 billion transistors, about 350 billion connections. That's a metric of the compute complexity of a piece of silicon. If we look at neurons, we look at what goes on upstairs in us, the human brain, we've got similar numbers of neurons, a little less than 100 billion, about 86 billion neurons or so. But the number of connections is vastly larger. It's about 600 trillion connections inside of this architecture, that's a few thousand times more connectivity than we have in a piece of silicon. And so it's really a very different architecture, and that leads to the ability to do some pretty extraordinary things. So I want to give you a little bit of history and background of our, yes, please.
You explain why connectivity is the issue. What's the importance of the connection?
I will, towards the end, I will say,
Not,
yes,
if
yes.
we don't understand that, then
Yes.
we don't see the picture.
Yes. For right now, we can assume that we have a few thousand times greater complexity in the human brain, meaning that the amount of information we need to use to describe the wiring diagram of the brain is a few thousands of times greater than the amount of information that we need to describe the complexity or the wiring diagram of a computer chip. Towards the end, I will make explicit what Rabbi Rappaport talked about, which is why that connectivity is very important for the kinds of things that we want to do in artificial intelligence. So let's go to one of the simplest organisms. This is an organism called C. elegans. It's a little teeny worm. This is, again, under the electron beam microscope. And this is about the size of a comma in a typical typewritten page. Very small organism. What's extraordinary about this organism, and why this is a model organism, is that every one of these worms has exactly 959 cells, which is kind of amazing. And more than that, it has exactly 302 neurons. Now that's kind of an amazing thing, because if you think about it, this thing is coded as we are by a genome. A genome is the set of nucleotides that sit inside of our nucleus. We have a genome that is about 3 billion nucleotides in length. That codes for us. It tells us how to go from a single cell to 2 cells to 4 cells, and to start to differentiate into different tissue types, and eventually how to create the brain architecture that we all have. So too with C. elegans, but C. elegans has a much smaller genome. It's about 100 million base pairs in length. It codes for not that many fewer genes, a few, about 20,000 genes or so. We have only about 30,000. But it's a much smaller genome that somehow codes for that entire organism, and specifically for this talk, codes for the exact geometry, the exact connected graph of how every neuron, all 302 of those neurons, are connected to each other, what the wiring diagram from a neuroscience standpoint is of this organism. And I'll mention that in just a second, but just to give some historic background, because this is quite interesting. This was a Nobel Prize. This was Sidney Brenner, who was the first one to decode that wiring diagram. And this was done back in the 90s or so. This is a worm that's fixed in a matrix, polymer matrix. And then what you do is you cut very thin slices of those. You take each thin slice, about a micron in thickness, put it under the electron beam microscope, take a picture that looks like this, and then you can build up many, many images like this. It's like a tomograph, like a CAT scan, but it's not CAT scan we're doing computationally. It doesn't have this resolution. Only by cutting these thin layers can you get this kind of micron resolution. And you need micron resolution because these neurons are on order of micron or so. And then you can computationally stack all of those up and recreate the wiring diagram of this organism. So this is kind of what this looks like if you take all of these images and stack those up and then use a computer to trace all of those neurons. We can now see exactly which neuron is connected to which other neuron. This is a way to represent this. This is all 302 neurons, and this is, for instance, this neuron. These are all the other neurons that it's connected to. This is the equivalent of what we call in computer engineering or silicon engineering a net list. This is the list of all of the different parts, all the different logic elements that are connected to each other. It fully defines the logical working of this organism. This thing is fully coded. So this is just a little fly through of this. This is a project called Open Worm, for those that want to take a look at that. And then let's see if I have a working video here. Oh, here we go. Not sure if I have sound to this. No. We can try your thing here. Okay. So here they took this net list, the wiring diagram of the worm, and they know which muscles it's connected to. This is called worm simulation. So this is a tool that you can play with. You can try making different connections. You can adjust different parameters and see how the worm moves. At any rate, so some folks recently, this was done, as I mentioned, more than 20 years ago. Recently, this last summer, this was done on a male worm, and since all the worms they looked at were male worms, they all had exactly 302 neurons, all wired up exactly the same way. They just figured that every C. elegans is like that. They never bothered to look at the female worm. Turns out they did that this last, yeah, totally biased, even in worms, right? So. That being said, they did do this past summer, and lo and behold, it's different. And this kind of triggered renewed interest in this organism. This was a group that took a robot, made a robot that is running the logic that's running inside of this worm exactly. So they have all of this net list that I said, all of the connections, all of the different neurons connected to the different muscles, and they use that to program a robot and to see what the search pattern is of a robot. This worm, much of its logic is searching for food, searching for other worms, and it's got a little photosensor, it's got an eye, and so they can code all of this into a robot, and now this robot is running a brain of a C. elegans and replicates a lot of the search patterns and things of, it's not terribly exciting, but it's a worm, so. But yeah. So it's got a photodetector up top, it's got a couple of motors, at any rate. The point here is that this thing is coded, meaning that worm, every single worm, we call this brain, by the way, a crystalline brain, because the genetic code in that genome fully codes for that wiring diagram. It's a one-to-one, it's extraordinary amount of precision, and actually a tremendous question. This is just a side thing, but there's actually a tremendous question of how that works. So this is a typical representation of how we look at genomes in general. So these are not all of the genes in the C. elegans worm. These are the genes that are related to neurons inside of the C. elegans. So these are all the neurons that are related to that. And this is a graph that shows the relationship between these different proteins. These are a gene codes for a protein. Proteins interact with other proteins called protein-protein interaction. And all of the logic, for instance, when we want to build a clock, a typical circuit that is built on every silicon wafer to test out the fabrication process of a silicon wafer is what's called a ring oscillator. That's a clock that works by connecting a transistor the output of one to the input of the next, the output of that one to the input of the next, and then tying that back to the beginning. And if you hook that up to an oscilloscope, you'll get a sine wave, and that's a clock. And that tells you how good your silicon process is. You can build exactly the same circuit in biology. And the way that you do that is you have a gene codes for a protein, and if you have that protein from gene 1 turn off gene 2, and gene 2's protein turn off gene 3, and gene 3 turn off gene 1, that looks exactly formally from a mathematics standpoint like that ring oscillator. By the way, once you can build that circuit, you can build any piece of logic that we can build in silicon. So there's actually a one-to-one formal correspondence between all of the things we can build in silicon and what we can build out of biological parts at the molecular level. So this is a diagram of all of the genes that are related to the neural architecture of this worm. And we have this because we can sequence the genome. We have this because we were able to take this slice of the into the electron beam micrograph. And a very, very deep question is, how does that genome code for that geometry? How do I get from a genome to this architecture? And one reason I like to know that is because I like to know how that works in us. We'd like to know how our genome codes for the complex architecture. It's not just the things that we learn, but when we're born with a lot of pieces of our neural architecture that are pre-wired up. We'd like to know how that code works. This is an undiscovered code. A little hint of how that might work is something called CAs, or cellular automata. If you think about the following, each of us start from a single cell. So we all start from a single cell. That cell divides into 2, that divides into 4, into 8. And at some point, and they're all running the same program, right? Because each cell, the whole organism, all of us, started from a single cell. And that's running the program that's inside of the genome. When that cell divides, it gets an exact copy of that program. So both cells are running the same program. Cellular automata is the mathematics of machinery that divides like that, where they're all running the same program, but yet nonetheless can create structure. And I'll just give you a little hint or flavor of how that works. So we know in developmental biology what happens is that when you go from a single cell to 2 cells into 4 cells, cells are sending out little signals to each other. They're sending them out by sending little molecules. So each cell sends molecules out and receives molecules. And so a cell can tell how many nearest neighbors it has. So if you think about the cell dividing, once it gets to about 8 or 16 cells, the cells in the middle have more nearest neighbors than the ones on the outside. And they have a little circuit inside that says, if you have more than this number of nearest neighbors, you're an internal cell. And now I can turn on some new genes that will make me a different kind of cell. I'll start running new programs that might differentiate me into cardiac tissue or neurons or things like that. That's a symmetry breaking along a single axis. There's a number of other symmetry breakings that go on, and this is how all the cells, even though they're running the same program, start to differentiate and create new structures. Really an extraordinary thing to think about, the fact that we all come from a single cell, but we create all the structure that we see. What we're really interested in is, how does that genome lead to the neural architecture that we see? Okay, so that's a little bit of an aside. So this is kind of where the field stood for about 25 years. So we had the C. elegans. It's got 302 neurons. As I mentioned, we have 86 billion neurons, and that's kind of a large jump. And the reason that the field kind of stood there is that really the only tool that we had to look at sufficiently high resolution where we could see wiring of neurons. We could see plenty of things in a MRI, or we can see things under an optical microscope, but we can't see them at the detail where we can actually create a wiring map of how that logic works. And so really neuroscience wasn't exactly static. There's a big program, a multi-billion-dollar program to do what we did for the C. elegans for the human brain or for the mouse brain, and that is to cut thin layers, 1 micron thick, put those under an electron microscope, take a picture, do the next one. But because an electron microscope has extraordinary resolution, but it can only look over a very small area, this was about a 30 or 40 year program, meaning like 30 or 40 years from now, we might be at the point where we've got the map of a mouse brain, not yet even a human brain. This is a very long, and this is an NSF-funded project. And a few years ago, along came my colleague at MIT. This is actually my undergraduate student in physics that went to Stanford and decided to switch into neuroscience. And the idea he had and his students had was just an extraordinary simple idea. Sometimes it's not yet a Nobel Prize, but stay tuned, I think it will be. Sometimes really simple ideas can give great advances in science. And the idea is this, is that anybody here who's had kids and changed diapers, you know that diaper has a polymer in it which has this extraordinary property that it can absorb a lot of water and change its volume substantially, on the order of 5 times or so. The volume of the polymer that's inside the baby diaper can swell and contain that much more water. It's really, that polymer network is a pretty extraordinary thing. And so the idea that they had is, let's take a brain, this is obviously a dead brain, and infuse it with this polymer, and then swell that brain, expand it. We're going to expand it by 4 or 5 times, and then that will make every structure in that brain large enough that we can look at it under an optical microscope. Microscopes are thousands of times faster. They can go over larger areas than electron microscope. And if everything expands exactly symmetrically, we should preserve all the structure. And here's some of the early results of this. This is a very non-dense part of the brain. These are just some neurons here. And you can just see that you just get a lot more resolution here. You can start to, even in this case, you could see the individual neurons, but this is not in a physiological setting. These are where things are spread out sufficiently to see, but you can see that you just get a lot more detail when you do this procedure. And so this has just been an extraordinary kind of advance. This is being used now for a whole bunch of different processes, not just neuroscience. This is cell division, for instance. This is all of the amazing machinery that happens when those 2 cells divide, as I mentioned. You have 2 copies of the genome that are coming apart, so the entire set of chromosomes are copied, and exactly 1 copy goes to the left cell, 1 goes to the right cell. There's a whole bunch of machinery, meaning proteins that are involved in that process, spindles that pull exactly at the right place all of those things apart so that everything goes where it should go. And again, these are processes that were just very, very difficult to see under an optical microscope. This kind of gives you a little bit of an idea here of, this is kind of the resolution that you had before, and now we just have much better resolution. And this was really not just better resolution, but it crossed a threshold that allowed us to recreate wiring diagrams critically in the brain. So this is now a big effort. This is actually from the Howard Hughes Medical Institute. This is a paper that came out this past summer, and this is the first complete wiring diagram for a fly. Flies are also very well studied. They've got vastly larger number of neurons than the C. elegans. This is really something that looks like a complex brain to it. This is showing this polymer swelling. You can see the size expansion that you get. I think they now can swell up to 10 times as large in several steps. So let me hope that this video works here. Let's see. I have to say, this is one of the most stunning papers that I've ever seen. This is just an extraordinary kind of scientific accomplishment. Simple in principle, but in practice. So this is the fly brain with its with its optical lobes in the front. Let's see if that looks like it paused there for some reason. This is all brain. This is all the brain. And this is just doing a fly-through. So again, this was done using a kind of microscopy called light sheet microscopy that can image planes of a sample at a resolution on the order of a few microns. But now when you expand this using expansion microscopy, that's sufficient to see all of the neural connections. Yeah.
How do you make sure that the expansion
Yeah.
process does not break any connection?
Yeah, that's a great question. So the way that that's done, so it does break connections. So these are not working neurons anymore. But what they do is they label the neurons with antibodies that have a specific dye to them that those antibodies only go to neurons. And so now when everything expands, the neurons are broken, but the tags, the color tags, all come apart in exactly the right proportion. Yeah. So they can trace it exactly, right?
Even if they are broken.
Even if they're broken, right, right. So you're absolutely right. I mean, this is not the same as mapping a brain that's living and operating. But there's many, many things that you can do here. For instance, those antibodies can tell you more than just that it's a neuron. It can tell you what kind of neuron. It can tell you what state that neuron was in when it was last working, and so forth, things like that. This video is pausing a little bit. At any rate, this gives you a little bit of idea of this. Now, just to give you an idea, that whole program that I mentioned was like a 30-year program using electron beam imaging to do a mouse brain. This can do a few flies in a day and a mouse brain in weeks period of time. And that's really important because one thing that we want to do for these higher organisms is look at not just a single organism, but we want to look at the variation. We want to, unlike a C. elegans where the wiring diagram is exactly the same, we know in us and in higher organisms that wiring diagram is different organism to organism. What we're really interested in is what is innate, what's innately coded by the genome, what's the starting architecture for these brains. And then, of course, when we learn new things, that wiring changes over time. Okay. Let me see. Okay, so this is, there's one other, let me just see if I can show you one other thing. Let's see, yeah, this one. So this shows, this is now zooming in. This is actually in mouse. So what I showed you before was a fly. I think we can do without that. This is actually in mouse. Now, this is not the entire mouse brain. This is a small little section of mouse. I think
Still being sliced.
this is not being sliced. This was a cube of a mouse brain, I think about 1 millimeter on a side, that was expanded and then put into a light sheet microscope where the light is imaging only on one plane, but it's not being sliced in a.
It's sliced optically.
It is sliced optically, exactly, right. Now here you can really see, I mean, this is, first of all, gives some idea of the complexity. This looks much different from a microprocessor where on average each transistor is connected only to a few other ones. Here you've got thousands of connections per neuron. You can just see the complexity of what this looks like. They did this as a warm-up to show that this is doable in a pretty high organism, which is a mouse. And this is really where they're headed right now. Actually, one of the first things they're going to do is back to the fly is, it turns out you can train flies. Flies, don't run mazes exactly like mice do, but they can be trained to go to certain things depending on electric shocks and other odors and things that they smell. And it's known that you can train flies. Can we see where that learning is resident, meaning let's take a whole bunch of flies, train some to do this, some to do that, and can we image those brains post facto and decode where those
The
memories are
flies'
stored?
brain are plastic, meaning they change the connection.
They are, yeah. The C. elegans is also plastic.
Really?
Yeah, the C. elegans is also plastic, but the wiring diagram that I showed and the wiring diagram at birth is those connections on a 0 to 1 basis, meaning is there a connection or not, that's static. Whether the strength of that connection can change to some degree, even in a C. elegans. Yeah, C. elegans are also somewhat trainable. You can give various shocks and things to them, and they're also somewhat trainable. Okay. All right. Okay, so where is all of this headed? So in parallel to this, as you probably know, there's now a tremendous resurgence in silicon. Most of this resurgence of interest is, almost all of it, is to power what are called GPUs, or chips that can run artificial intelligence algorithms. And to do that, you need, as Rabbi Rappaport said, you need many, many connections. You need a different topology than what we typically have in a linear microprocessor. We need big matrices of numbers that are all connected with many connections to other big matrices of numbers. And this looks like what's called a fabric. This looks like a connection architecture, which is much more, and the reason we use the word neural network, much more like what we have in the brain than what we have in normal pieces of silicon. Now we can run these algorithms on a typical architecture in silicon because you can do it piecewise. You can do it a little piece at a time. If you only have a small number of connections, it's slow. And so a lot of the great results that we've seen in AI, for instance, things like folding proteins that came out of Google's DeepMind, or recognizing cats in videos, or all of the things that we somewhat associate with humans, many of those results are run by using tremendous amount of processing, meaning thousands and thousands of processing cores, millions of dollars of processing in order to be able to do that. So recently a company, this was also announced over the summer, announced a chip that is the largest ever built. This has about more than 1 trillion transistors on it. So I mentioned that the biggest chips that we had built to date are about 100 billion, about 50 billion transistors. This is this is 20 times, more than 20 times larger than that. And it also has a large number of connections to it. Now this is still about 10 times fewer connections than what we have in the human brain, but it's starting to approach the kind of connectivity that we have in the brain. I won't go into
Many more transistors.
what's that?
Unbelievably more transistors.
Unbelievably more what's that? Yes.
1 to 1 million transistors, much more
Than we have in our brain, right. So this has many more transistors than we have in the brain, and because of that, we're starting to get to the total connectivity complexity that we have in the brain with a chip like this. You might ask why this, by the way, this is the largest chip that we make to date, just in scale here. This thing is much larger than that. When we make chips, we make them in big wafers like this. They're about 300 millimeters on a side. They're made in what are called a fab, which is a factory for chips. It's the place where they run the photolithography process. These factories also obey a geometric scaling, meaning every year they're getting geometrically more expensive. The most expensive of these fabs now is more than $10 billion. The reason that this wasn't done before is that it's just very difficult to guarantee that all of those connections can stay in place. over such a large area. The reason that our cell phones and our memory disk drives are so cheap is that when you make a big wafer of these things, you don't require that all of them work. In fact, they typically make a wafer, and then after the fact, they test which ones work. The ones that work get shipped. The ones that don't work get thrown out. If you put enough money to make all of them work, this would be a really inefficient thing to do. It's much more efficient to run that fab quickly with some error rate and then just test the devices, whether they work or not, and ship the ones that work. And then even within an individual device, like a memory device, they do a very smart thing, which is that even in one device, not all of the transistors are working in that one device. They have lookup tables that are look at which of those transistors are working, and they make a map to the working transistors so that any memory that you want to store on this is fanned out to only the working transistors. And so you get a very large yield. When it comes to microprocessors, we haven't been able to do that very well. So memory, we can make a map to where the working transistors are. In a microprocessor, we need every transistor to work. And that's why we've been limited in the scale of how large a microprocessor we can make. That's why we've been limited to on the order of 50 billion transistors or so. And the advance here was to figure out a new architecture where, again, it would be impossible to make sure all 1.2 trillion transistors work. But what they did was to say, we're going to use 10% of our transistors to be able to wire around defects. And so when there's a broken transistor, there'll be a nearby transistor or set of transistors that we can wire around that and still have a working device, even though we've got some broken device.
So the structure can
Yeah.
change.
It does not. It doesn't change physically, but it can be reprogrammed on the fly to run around those. Yeah. It automatically detects by running test algorithms and of where the defects are, and then routes around those.
But the function
Yeah.
before and after the fix
Yes, same.
is going to look the same if I quantify
Right. The function after the fix is as if I had a perfect wafer to begin with. Correct. Right. The cost of doing all of that is about 10% of my transistors are involved in that rewiring.
And some other percentage are not working
Yeah.
at all.
And about 1% are not working at all. Right. Right. So about
parallel process in our brain.
is there a parallel process in our brain?
Which means
Yeah.
these errors happen in our
okay.
brain.
Yeah.
Neurons,
Ah.
they transfer the information from one neuron to another, and then you can get a more efficient network rather than
Ah.
just like regular neurons that are specified for a certain function.
I think what Robert said is if we have some neurons that die, or
Right.
can we wire around that? That's the question.
Oh, yeah.
Yes.
It's called Schwann cells, if I'm
Uh-huh.
not mistaken.
That's what happens with strokes when people recover from it.
And it seems that the plasticity in young children, if you were deprived of oxygen or somebody, God forbid, was, underwater for a while, that plasticity is much greater in young children, that ability to rewire. I don't know the details. I'm not sure if maybe Rotem knows. I don't know the details of what that rewiring architecture looks like compared to the way that it's done in silicon, and what the overhead is. Here again, 10% in this example I showed you here
Dedicated to that.
is dedicated to that rewiring. Neurons, not sure.
And it's physical or
Yeah.
What do you mean?
the change is physical? Here it's
It is
logical.
physical because
Our brain
the
is physical.
neuron just passed let's say the other damaged neuron, and
Yeah.
creates a connection within the neuron in which he has to create a new connection.
Yeah.
So God did a
Yeah.
better job.
But you
I'm-
cannot apply
That I'm
this-
sure.
You cannot apply this to every single neuron. Sometimes when you have a defect, that's it. It's gone. So it depends.
Yeah.
Very long ago, I saw a paper saying that even new neurons are produced in the brain. Not just there are new connections between existing neurons, but even new neurons, new cells are produced as well. So new neural tissue can be grown.
I think that that's correct. Certainly young humans, that's true. How well we can do that
Yeah, I'm probably
in
on
older,
beyond
right?
that now.
Yeah. Yeah, right.
Young children, philosophy.
I better take care of myself.
Right. So a really interesting question. This is something that's been looked at is, what if we think about these two fields coming together? So I mentioned to you the C. elegans, 302 neurons, trivial to simulate, as you saw in that robot. The fly that's got a million or so neurons, that's also simulatable with today's silicon. What about the human brain? And so if you look at the complexity that we have of the brain, the numbers that I showed you before, about 600 trillion connections, about 86 billion neurons, that right now is a very expensive thing to simulate. That would probably be on the order of hundreds of millions of dollars, not impossible, if we had the map. But if you think about the next 10 years, both what we'll have from a expansion microscopy standpoint, certainly in the mouse and potentially in the human, and where we'll be from a silicon standpoint, the kinds of chips that I just showed you, in about 10 years from now, or this is 9 years, for about $300,000 of compute, you can go to Amazon or AWS, Amazon Web Services, and buy as many cores as you want. If you want to spin up a big computational process, you can just go rent CPU cores for as many seconds as you want. So for about $300,000 for a single second of emulation, we will be in a position to emulate 600 trillion connections across about 100 billion neurons. That's a pretty interesting and kind of exciting data point, I think, that not too far from now, we should be in a position both on the wiring diagram standpoint and on the computational ability to simulate that standpoint to start to simulate the circuitry that's taking place in the brain. Now, there's lots of things that are still unknown. For instance, we don't know the exact transfer function of all of the neurons. We don't know the exact mathematical equation of how those behave. And there's many different kinds of neurons. But presumably, we should be able to decode some of that also with expansion microscopy. As I mentioned, we can tag different neurons with different antibodies. We can light up what state and what type of neuron those are. And presumably, there's some regularized set of rules of the transfer function of each of those. We can encode that into the computer simulation as well and start to simulate some of the things that are going on in the brain. This, of course, brings about in sharp detail a famous question, which is, just like the C. elegans that we can clearly see, 302 neurons totally coded from genome to those 302, that connected graph of the neurons, that is a totally coded thing. We would be hard-pressed to say that there's anything that resembles free will or free choice in that. To the best of our knowledge, certainly to the best of my knowledge, the same appears to be true in higher organisms as well. We've seen it now in a mouse for the first time. Haven't yet done the human brain, but the human brain, as far as we know, does not appear to be different. Now, this is not to say that there's not stochasticity, there's not randomness in those neurons. It's the same is true in a computer chip as well. Every computer chip has a threshold that's set for what the error tolerance is. Typically, we set our error tolerance to a part in 10 to the 12. About once in a trillion times does a microprocessor make a bit error. By the way, this is well, I don't think I'm giving out any great secrets, but one way to crack computer chips, let's say you have a bank card or something, is that if you turn down the voltage on that chip, the number of electrons that represent a logical 1 goes down, and the error rate scales as the square root of number of electrons, as Poisson statistics. And so if you tune down the voltage and start looking at the errors that come out of a computer chip, you can back out the RSA codes, the encrypted codes that sit on top of a computer chip. Any physical system has errors. It's just a question of what that error threshold is, and can you do error correction on top of that. And every logical system has a layer of error correction that sits on top of that. So this is not to say that the brain is totally predictable. It's a physical system like any other physical system. It has fluctuation to it. Same is true in a computer chip, but to the same degree that we would say a computer chip doesn't have free will, there's nothing that's not simulatable to the best of our knowledge. From everything we can see and know now, this appears to be the case for the biological neural systems that we're now looking at in very fine detail. Let me say a little bit about some.
There are some philosophical questions
Yeah.
there.
Yeah, there are some very deep philosophical questions. Yeah, I'm going to leave that to others, I think, to discuss. But if people want to bring that up, I'm happy to engage it. But absolutely, right? I mean, the biggest one is that as a society, we tend to punish people that do the wrong thing. So how can we do that if they're totally wired and?
I thought
Yeah.
we would just come up with
What?
Socrates' argument for the
Yeah.
materiality of the soul,
Uh-huh.
the spirituality of the soul from the freedom
Yeah.
of the will. And it is only a free will that,
Ah.
that can be held responsible morally. So that is why it is an important metaphysical and moral issue.
If the
Obviously,
yeah.
on top of all these technicalities.
You're only looking
Yeah.
at the material side.
Yeah.
So you would expect this to be deterministic. But if there's an immaterial
Right.
side, which of course there has to be if
Yes.
there's free will.
Yes.
Yeah,
Which
okay.
obviously
So-
there's free will. So.
Okay, we should shut up.
Yeah.
Well, you would say that.
I also believe. I also believe in free will, as my daughter-in-law can attest. Every Shabbat dinner, this is our favorite topic to talk about, because we're not allowed to talk about science on Shabbat, so, or at least. So that's. Anyways, what's that? Yeah, we can philosophize it. Yeah. Let me just make a few more remarks. Tell me what my timing is, Robert Repo.
So 6:20.
Oh, I'm going to be well within that. So okay. I just want to tell you about a couple other related topics that I think are really, really interesting. This is some work to look at fMRI. fMRI has been used significantly in a whole range of things in neuroscience. MIT now has a class in their neuroscience department called Neuroscience 9.46, which is fMRI and ethics, something like that, looking at ethical decisions in an fMRI context, looking at the brain pattern in when people are making different ethical decisions. This was a kind of cool paper. This is a paper that came out of the Neuroscience Institute in Kyoto that got a lot of press. And the idea there was, could I, just in fMRI, decode something that you're thinking about? And the way that they did this was to start by training a neural network, and I'll try to explain exactly how this works. But typically in a neural network, what we do is we have a whole bunch of associated things. For instance, I can go on Google and look at Google image search. So I can look under animals. So I'll have cats and dogs and birds and lots of things. And then in the neural network, I'll try to associate all of one kind of thing, like cats. So I'll label all of those images, and everything that's a cat, I'll tell the neural network, this is a cat, this is a cat, this is a cat. So that's what we call a training set. And if we build a neural network, as I mentioned, is a set of connections between that input data, meaning the pixels in the input image, and a output, in this case, a binary classification, cat or not cat. And that's a big matrix of numbers that I try to connect. Presumably the way the brain does that is that you've got a large number of neurons that can all change their connection strength to some output. And after enough training, a human can classify what's a cat, what's not a cat, and so too can a neural network that's got a camera and it's looking at an image, or it's being fed in images. So what they tried to do here was to start by showing you the letters of the alphabet, and so it knows what letter the person is seeing, and then take an fMRI scan of that person's brain when he's looking at the letter A. Now, if you look at that fMRI scan, that's very tough to see anything there. It's a very complicated, not spatially mapped image. But if you do that enough times, so you show the person a letter A and show it to many people, the letter A, and memorize all of the different patterns in the fMRI activity that correspond to A, and then do that with B and C and so forth, this is the result that they get. Once they train that network, now when they show a person a new A, or a new random letter, just purely on fMRI activity, not knowing anything else, these are the images that they create of what the person is seeing. And this is rather remarkable. There was previous work to train a neural network to know whether you're looking at an A, a B, a C, or D. That's already interesting. But here it's actually recreating the image of what you're looking at. So that you can see that these are not, that's not a perfect A, or that's not a perfect C. But it certainly resembles an A, a C, an E, an I. Those letters certainly have some resemblance. Now, when you try to go to a more complicated space like this, we're actually showing the input of a picture, like an animal. So here's what they're doing here is they're reconstructing what you're looking at. Now, this is less perfect. This is not quite of the same quality. But this is a fairly remarkable result, that you can get that just by looking at fMRI. Obviously, this has even the alphabet has some wonderful consequences for people that can't type or can't paralyze. If you look at a chart of letters without any electrodes or anything, now fMRI is an expensive piece of equipment, but
Have they tried to do it without the visual image?
Yes,
but just
exactly, right. So that's really where this is headed. Now, I didn't put the video here. The goal of this is to try to be able to decode things that you're thinking of mentally without the input image. You can go online and look at the results of that, and I think there's debatable whether there's real signal there. You see images, whether they correspond to anything that the person is actually thinking, that's a question. But that is the goal. Could you decode dreams? Could you decode visual things that I'm thinking about with my eyes closed?
But even
That's a
this
great
would
question.
just be what
Yeah.
you were imagining.
Yeah.
So you'd be
Right.
to move to something more abstract.
Yes, yeah. It would be a pretty extraordinary thing. I on balance, my threshold is fairly elevated for being convinced of anything. So I don't think that has met that threshold yet. But that is what they're trying to do. Yeah.
That would be just imagination.
Well,
not
exactly.
really thinking.
I agree with
Yeah.
you.
What's that?
Not thinking, but
Yeah.
we just imagination.
We have to
And,
talk about
Right.
actually it's
universals.
just
What's that?
We have to talk about universals.
About
Well,
universals.
we'll get
Okay.
there on that.
Yeah.
Okay.
All right.
I actually was very motivated
A cat.
earlier on. What's that?
Cat.
What do you mean by
is
cat? What is cat?
Can it be visualized?
E-
Or better question.
Well, certainly if you're dreaming, you have a pretty stark visualization of things.
Yeah, but
Whether you can do that.
Triangle.
Yeah.
What shape is it? 3 sides equal? 2 sides equal? No sides equal?
When you first said the word triangle, I said 3 sides equal.
That was
but
your image.
That was my
Yeah,
image.
was
Yeah.
that triangle?
But that's an image, but it's not the concept
Yeah.
of triangles, all triangles.
So you
Including
probably know, I
scalar
spent a summer in Leon Cooper's lab. Leon Cooper won the Nobel Prize for superconductivity, but then he went into neuroscience later in his life, and he was looking at the LGN, the lateral geniculate nuclei, which are the higher circuits so you've got the visual cortex, and those plug into a layer in the brain called the LGN that are shape detectors. So these are things that fire differently if you put a circle or a triangle or a square, or if you have any shape that's moving left to right but not right to left. These are all of the primitive detectors. There is a triangle detector. There's a 3-sided detector there. I forget whether it needs to see an equilateral or it needs to, I forget what the boundaries are there, but yeah.
But this process,
Yeah.
On the technical level, this does not follow connectivity. It follows, blood flow to areas in the brain.
What is fMRI looking
Right.
at? Exactly.
So it's not, the resolution is always going to be not on the level of connectivity, and even on the level of activity, it's going to be,
Yeah.
it's going to be looking at what? At the axons? I mean,
So, right, the resolution, so fMRI, Rolly's right, fMRI is looking at glucose uptake or glucose metabolism. Its resolution is probably not better than hundreds of microns. I don't know if you know the resolution. Yeah, but it's not single neuron. Yeah, it's hundreds of microns. So we're talking about hundreds of thousands of neurons in a single voxel of fMRI data. That's correct. Yeah.
And
Yeah.
that may also account for the blurriness of the image.
So I'd have to tell you more technical detail of how this is done. This is the way that neural networks are set up is this. The way that this is done is to feed in a finite set of images. So this might have, 1,000 different animals in it. This does not have more than that. And it is now trying to determine which thing you're looking at here. This works, this only got fed in the letters of the alphabet. This has, a few thousand different animal images, and it's trying to reconstruct the image you're now looking at. This is what this is doing. This is a simpler task than trying to do, and all of the work in this area has done this. This is a simpler task than trying to create a pixel by pixel image of what you're looking at. What's different here is this thing doesn't look like any image that was fed in. This is what's called a GAN, a generative adversarial network that is generating a new image. But the basis set that it can generate from is not the set of all possible pixels. It's the set of images that can be composed from those 1,000 initial images. That's why this looks like something. I don't know.
Bear.
Yes. What's that? Again, yeah. I looked at this page. We can look after this. They have a whole bunch of examples. Some look convincing. Some don't look convincing. All right. So I just want to end just with and I apologize for the folks that were here last time, but this is now becoming kind of an even more pressing topic for us, which is what are the rules and the guidelines by which we imbue our now thinking devices with the appropriate ethical frameworks that that need to be there in some form or another. And I'll just, we actually started a program recently, my lab, not my personal lab, but the MIT Media Lab with Harvard Law School, to look at ethics in AI. This was funded by Reid Hoffman, who's the founder of LinkedIn. And this is just driven by a whole bunch of examples. One really salient example is that you may know now that across many states, sentencing guidelines are being set by AIs, by artificial intelligence. So what do I mean by that? We can look at the sentencing parameters. You know, when somebody's convicted in the U.S., there's still latitude to the judge as to, do they get 3 years, do they get 5 years, first offender, what are all the parameters, where do they grow up, how remorseful, all of those things. You know, there's a guideline, there's a set of guidelines of what sentence this person will actually get. That has typically and always been done by a human judge. Caseloads are heavy, and you can't put enough thought into each case and enough consideration in each case. And so what people are doing are looking at the entire case record of every sentence that's been handed down over the last 50 years in the United States, state by state, district by district, what the offense was, and what sentence was actually handed down in that case, and what the result was. Was there a repeat offense after that? Did things go well? You can train a machine learning network to take in a new case that it's never been seen before, and the sophistication of this is pretty great. You can actually have that machine read the case file. So you don't have to type in all the individual, it actually can read the case. That's kind of an amazing thing. At the end of that comes out a guideline for the judge. Now, that judge has a stack this high. He can either pour through this case with hours and hours, or he can look at the recommendation of the computer. Likely he's going to look at or she will look at the recommendation of the computer more and more. And so this is just one of many examples. And the question is, how do we, so the way that is being tuned or set is based on stare decisis, on precedent. Is that the right way going forward? Or maybe by using a machine, we could do better than that. Maybe by a machine, we could say, all of these cases, the machine now has discovered that all of these cases where we put a very harsh sentence, that didn't work out very well. That person ended up learning bad things in jail, repeating, whatever, certainly in this district, in that district. Maybe we can do better. What's the right way to set those parameters? This was an effort that's pretty famous and well known. This was a colleague of mine, Yad Ruwan, who had a different approach, related approach for setting the ethics in self-driving vehicles. And this is a really famous program. You can go online and try this out. This is called the Moral Machines Project. This is published in the journal Science. And the idea here is we can do no better than to crowdsource our ethics, rightly or wrongly. And I'll tell you a quick story in a second about Iyad. But he comes from not too far from here, over the border in Syria. But his idea was show lots of scenarios, meaning have people vote. Should this driverless car with no people in it stay straight and kill these 3 people, or swerve? Is no action, killing the same number of people, no action better, or a positive action better? Is there any difference in those? Whole bunch of scenarios. There's a driver in the car. That driver is going to kill 3 people and a dog, or it can swerve and kill the person and save these people. Which one should it do? Now, many people would say it should swerve because it's 3-1 and a dog. Some people say that's 4-1, by the way. You're surprised, but I'll show you. 10% of people see no difference between saving a dog's life and a person's life. And it varies by country. From the yeah, right. Well, okay, that's your opinion, but okay. But if you think about this is not such a trivial question, because if you know this car is going to kill you, you might not elect to drive in this car. So the idea is self-driving cars will be safer,
Right.
but only if people will use them. And if you know that it's programmed to kill you, the uptake rate might not be very high. And so these are the statistics. This is as of like a year ago. This is about 40 million different decisions have been made all over the globe. You can parse this out into different regions, different people vote differently on things. So, corporal punishment, in some countries you're a bad parent if you don't whip your kids for not listening. In other countries you'd be arrested for doing that. So is that right? Is it wrong? These are just what do you guys say? Hey, I'll show you this.
This section by saying you could do no better than.
You could do no better than crowdsourcing ethics.
Yeah, in what sense could you do no better than crowdsourcing?
I'm not saying that. The presumption of this website is, let's say this was a project that was originally done for Google. Google wants to minimize its legal cross-section.
So you could do no better legally.
Yes. Right. You can do no
I just
better.
wanted that to be explicit.
You have no better defensible position in a court of law than to say, you know what, I asked 10 million people and the majority felt this is the right ethical thing to do. That seems like a pretty strong foundation to stand on.
It shows Asia and
Yeah.
About the
Yeah.
judge advisory program.
Right.
Judge ed program. You need, for artificial intelligence, you need a specific result. Like, you try to have less repeated offenses. Right?
Yes, yes.
Very focused. Yes, about volition. A judge can decide that even the dog is 95 years old and is not going to repeat his offenses, still you should put him on an electric chair because justice demands it. Not because he'll repeat and kill more people. He's too old for that.
Okay.
So
Yeah.
this artificial cannot measure, cannot imitate.
Well, unless it was a case that had never come up before, it can see all the precedentary cases of all the 95-year-olds that have come before the court and what sentence they got.
If there are few cases like
Yeah.
this, it won't work. You need
Yeah.
thousands and thousands of cases like this.
Or maybe it can extract. It can see that as you go from age 70 to 80 to 90, you're starting to fall in the amount of sentencing you get, and you can extrapolate that curve. AI can do things like that. It can learn a function. It can fit a
But
function
still,
too.
it doesn't.
Yeah.
Okay, so it measures what people do,
Yes.
but we are far away from
Yeah.
thinking a new thing or starting the value of justice.
I agree with that, unless by looking at enough cases, those things become one and the same. Meaning, our ideas of justice and what we've done over the last 200 years or 400 years in England is itself a full enough set that the AI could learn something that we would call justice from it. That's a question.
Unless you say that the
Yeah.
judge, in the individual sense of
Yeah.
justice, is not obligated to his fellow judges, so we're talking free will. So a judge can
Yeah.
decide as justice is very, very important, much
Yeah.
more important than the thing of offender, and some people
Yeah.
don't. So
And-
here is the
yeah.
difference.
But presumably, if you looked at 400 years of case law in Great Britain, for instance, which we have, you could see the distribution. We have some judges that justice is very important, some justices that are very practical, and there's a whole distribution. And by the way, over the population, what is that distribution? And let's have an AI that mirrors and reflects that distribution. And it's changed from the 1600s to 1700s and the 1800s, and we can see how that correlates. And yeah.
There have been a lot of articles recently about how certain algorithms have accidentally codified bias
Yes.
into the process.
Right. Eh.
So is there an algorithm also to correct that? Right? I'm
Well,
thinking if you're
that's an awesome point, because that's exactly the problem. If you train, in the United States, on the last 50 years of sentencing guidelines, black males are going to be sentenced longer because that's what we've done. And so how do you, as Rabbi Rappaport said, how do you ever get out of that? How do you jump out of that paradigm?
So you have
That's-
to alter the system to ignore that, so, but it's
Yeah,
not a
yeah.
system decision.
Yeah.
It's human decision from free will to dictate to the system
thing
to
is-
ignore the race.
At least there's a school of thought that would say that yes, you need humans to set overarching guidelines that are unlearnable from that. Now, it's not that is definitely a prevailing thought. It's possible that if the AI could suck in enough data, it could see that this hasn't worked very well. You know, we've sentenced black males to much longer sentences, and if we look at across that whole distribution, the ones that we set to shorter sentences, they actually did better. It might be able to learn a rule.
I could care less
Yeah.
about it. I think that black
Yeah.
people are inferior. So they should get tougher sentences, even if, again, humans have to decide what we try to achieve. And this is pure free will, pure crazy. If I think
Yeah.
that black people are inferior, black
Yeah.
people are equal, it's absolutely free will. And
I-
the ethics, it's nothing to
I
do.
agree. At some point, we have to say what we're trying to globally optimize for. Are we trying to globally optimize for economy, for a better economy? Are we trying to optimize for jobs? Are we trying to optimize for individual contribution to society? I agree with that. Though we're setting what makes a good piece of artwork. That is a human thing. Computers that can then copy what we think are good artworks and create new artworks in that template, they can do very well, right?
I was just returning to this crowdsourcing issue and how this popular vote should decide
Yes.
what the moral basis of sentencing
Yes.
in these, or judging in these cases would be. We tend to say that people usually vote with their pocketbooks.
Aha.
And by that standard, let's say, Elton John would be a greater composer than Beethoven,
Aha.
because people are willing to shell out $20 per CD for Elton
Aha.
John, and
Beethoven less so. Yeah.
Beethoven does
Yeah.
much worse
Right.
in the music
Right.
stores. So
Right.
you can get out for cents, a couple of cents, or
But if you talk about
just from the basket for free. It's just lying
But
around.
if all you're trying
So-
to do is to minimize the legal liability.
So and that is but the point is that
Yeah.
popular opinion may not be the best guide in these.
Look at all the bad elections that we've
value
had over the
issues.
last.
Yeah.
That's certainly true. Right?
Yeah.
All right, so I'll just stay on with these last 2 or 3. What do you guys what do you say here? I'm going to show you the actual votes globally.
Mhm.
This is go straight, kill the passengers.
Yeah.
Most people, that's the ethical thing.
No, you have to.
Oops. Okay.
You have to kill the driver. Yeah.
You have to kill. That's go straight. Yeah. 61% goes straight. Little kid. There's nobody in the car. Kill a little kid or kill the adult.
Kill the adult.
Kill the adult. Swerve. Yeah. Okay. Okay, this is a good one. This guy is in the. This guy has crossed.
Median.
This guy has crossed the median when he is not allowed to, I think.
Definitely kill him.
And he's
No,
wearing
no.
an annoying hat.
Actually, I'm not sure.
He deserves it.
No, I think right, the light is green. This guy is here illegally. This guy is here legally. Right. So kill the crosswalker?
Yeah.
That is a very severe offense. Gotta kill him. Let's see, swerve. Yeah, 72%. Kill the jaywalker.
Philippa foot is rolling in the grave.
But again, it has,
How about here?
It's
Kill
not
two
the-
that are jaywalking? or kill one that is
Probably
in the
two.
wrong,
Probably
what's that?
That's the Canadian solution.
Did it try to ask if you are the
Like that's
person,
corrupt.
Yes.
you are the person that's corrupt.
Yeah, it
Who
has
do
that.
you
It,
prefer
it,
the
ha-
driver to
it,
kill?
it has that. It has
It
that.
was
Yeah.
that, it was
Yeah.
that.
Yeah. So this is you and others. So here,
Oh, good.
here, saving more lives. If we're talking about other people, not me, I definitely want to save more lives. If it's talking about me, yeah, a totally different answer. Yeah. Right. And here's the one that I mentioned here. People and pets. There's about 10% of people, I think I have it quantified here, that the way this works is that the more to the right, the more you favor this
Yeah.
parameter than this
Yeah.
parameter. So most people favor sparing humans, but there are, I looked at the actual raw data, there's like 10% of people that, no difference. They're both living things and that's it. So, anyways, so this is pretty interesting, and these are, this is at least one going ethos of how we're going to program machine learning things. We're going to take a vote, and it's probably driven by this legal cross-section argument.
No one will dare to ask a non-politically correct question. It's a black or white. They won't dare ask something like this.
Such as,
You have
Yeah,
a black
one black
and a white
person or one
Uh-huh.
white person?
they will not answer that question here, yes.
So not ask. Or
Well, they
someone
ask
is
me,
invalid or
they do ask gender here. There is gender. This doesn't mean most people prefer sparing males. This means no difference. So there is a idea that it's more important to spare women because they will carry on the next generation. And so there is a
That's why we
slight
have an
bias.
all-male
No, that's male chauvinism right there.
No, that's why we have an all-male draft.
Why? Oh, yeah.
Because you can't reproduce the
Chivalry.
next generation if you
Exactly what I said. You're just repeating.
So you just have a mine shaft cap to contend with.
This is should fitness. How about fit people versus large people? Should there be
Spare the
the
fit.
spare the fit people? Not sure why. Oh yeah. Here's
Is there a large scale competing paradigm to this?
what's that?
Is there a competing paradigm to this that has funding on the same level?
Not that I know of how to source ethics.
Yes.
Not that I know of. Not that I know of. So that is a great. There's actually hidden in what I said. There's actually two paradigms that we do have. One is, do we crowdsource from millions of people, knowing that people are not all that thoughtful and haven't read case law for the last few hundred years? That's this.
Right.
Just go source public opinion. You go onto Amazon Turk. You know how to hire a million people tonight if you want to. You go to Amazon Turk, you pay them a penny per click, or this was 25 cents a click or something, and you can get 1 million votes, 10 million votes, as many as you want. That's the crowdsource. Model 2 is stare decisis, which is looking at all of case law, looking at what considered trained judges in different countries have done. Those are the two paradigms I know. And
Well, and then trying to classify
Yeah.
what the relevant data points are to relate the decisions to.
Yeah, which is
And
most
then write your algorithm to be able to pick these right.
you mean how to choose these scenarios? Is that what you're saying?
Well, no,
Or-
if you do stare decisis
Yeah.
and you say, okay, X number of judges facing circumstances A,
Yeah.
B, C, D, and E, but
Yeah.
but somebody has to decide, well, what are
Yeah.
A, B, C, D, and E going to be
Yeah.
that are relevant
Yeah.
for our understanding the significance of these decisions, right?
Correct.
So if you
Correct.
overlook
Yeah.
relevant factors,
Absolutely, absolutely, right.
These
There-
are very
Yeah.
abstract scenarios, actually. So
these scenarios?
yes, and these scenarios are very abstractly set up, and
They're not
they may
that
give you
abstract,
some
I
idea about
Yeah.
generic
Yeah.
guiding principles, but perhaps the actual precedent
Yeah.
would contain information, much more concrete, relevant information about the scenario than these abstract pictures provided. It's, oh, you have two choices,
Yeah.
go straight or swerve. And
I see
you
what
only
you
have.
mean, this
you
is
only
just a
have
binary
whether
deci-
it's
this is a
male or
binary
female
decision,
or
but there might
etc.
be-
But
might be multiple,
That is
yeah.
precisely what
Yeah.
involves the prudence of
Yeah.
the judge.
Yeah.
Or the equity of the judge.
Yeah.
That is the Aristotelian.
Yeah.
But what-
for that.
what-
And that takes into account a lot more concerning the actual singular circumstances. So it is
Yeah.
not just having universal principles and do our calculations and see out the result of the sentence. The judge takes into account all sorts of particular circumstances in the
Yes.
case then, that cannot be coded into these general rules. So it is not just the general rules. And
So
that's
the other thing
precisely why-
is, the other thing is, okay, in
Yeah.
the self-driving car thing, you're just deciding what the car is going to do. You don't have any information on whether the people see it or what. I mean, an actual driver always is
Yeah.
gonna
Yes.
at least initiate a swerve.
Yeah.
Yeah.
But then the car is processing information much faster, probably.
The car is processing information vastly faster. It's got a 360 look around, and
which the driver doesn't.
which a driver doesn't.
Right.
And but more than that, this is Google, so they know umpteen million things about who's driving that car, and they know umpteen million things about all of these people that are in the road too, because they've got facial recognition. They can go in and say this person's in this social economic,
Right.
status. They've been to jail 3 times. They've
Voted for
contributed.
Trump.
They voted for Trump. They. Let me just. I'll just.
John, did you want to say
Yeah.
something? John's a lawyer.
Much better.
This was it was a simpler case, but I
Yeah.
did a case in the Supreme Court one time. The Social Security disability system is the largest adjudicate agency in the Western world. And in the 1970s, a professor at Yale designed a system for deciding whether people were disabled or not for purposes of getting OASDI benefits. Aside from some disqualifying conditions, automatically disqualifying conditions, there was a grid designed with age, education, work experience, and how much weight you could lift.
Oh,
And you just found the proper box on the grid, and you were disabled or not. And so this was, all the courts of appeals in the United States were divided on this, so I was representing the Social Security Administration in the Supreme Court. The question was whether this was okay or not, and the court decided that it was.
That it was okay.
It was okay.
Having that's fascinating.
That is really
Fascinating. Yeah. I'll say one last thing. Stare decisis, looking at the set of case law, crowdsourcing, getting everybody's opinion on an equal footing. There's one third possibility that was suggested by Rabbi Jonathan Sachs, which was game theory. That in principle, with enough simulation, you could simulate the outcome of different ethical rules. Now, that's a very difficult program to
No,
execute.
but then that still
Yeah.
requires you to define what is an optimal outcome. In game theory,
Right. This is
an
this is
optimal
this is Rabbi
outcome
Rappaport's
is
point. Is
economical.
humans have to define what is.
And then still it just pushes the
And they
envelope
can
further
change.
to, but
Yeah.
it's
The
still.
thing is, a human
Yeah.
decision can change from day to day. If I bet the Catalans show
Yeah.
that before lunch I think this way and after
Yeah.
lunch. They made the research about the decision depends whether it was before lunch or after lunch. And this,
Yeah.
in order
What?
to program all this in a human
Yeah.
decision to factor it, so it's really it's infinite cycle. It's a vicious cycle that you can never take control.
On the other hand, we know that technology is vastly outstripping the rate at which we can create new laws. And so we need some way to bridge that gap. We need a system to be able to handle this. Otherwise, we will find an
So
infinite
maybe
number
it's
of
better-
loops around the law.
I agree that
Yeah.
it might be, you say, better than human decision.
Yes.
Because better to
Yeah.
ignore the lunch thing, but human
Yeah.
people do.
Yeah.
But it won't be human decision, it will be something totally different.
Right.
Well, I mean, one might hope that actually self-driving cars, precisely because they have so much more information and quicker reaction times, would in fact be in these situations less frequently
Actually, insurance companies have already adjusted for that. Liberty Mutual has planned for fewer accidents when self-driving cars come
Really?
online. Yeah, my son is their general.
And the- and
general
there's a
counsel,
solution
but
to
yeah,
this.
they've already
for-
figured
really?
it out.
One solution.
I hope
to
they're
this-
right, but
Yeah.
yeah.
One
On
solution
the
is that
other-
Google's
I can
always
count on
liable,
Yeah.
that if they're gonna get into this
Their
business,
bottom line.
Yeah.
there's a standard called strict liability, and if
Yeah.
the accidents will be lower, and Google will just have to pay for them.
Yeah.
So there won't be any incident, I mean,
Right,
that's one possible
Yeah.
legal
The other
response
hand,
to
yeah,
all of this.
as you know, those actuarial tables aren't always right. GE
Right, right.
almost went bankrupt because they did not anticipate how long people are gonna live. And,
May I ask you a
Yeah.
very crazy question?
Yes.
And a stupid, what is your research actually? You showed us so many different perspectives.
Ah.
Now. I just want to know what you are doing now, these days, what your research is.
It has very little to do with any of these topics. What- my lab runs a molecular biology lab. We work on CRISPR, genome editing of the human genome, and we work on artificial intelligence tools for designing drugs, small molecule drugs to protein to cancer targets. Those are the main areas that we're focused on.
Thank you.
Yeah. Thank you. Great. I talked about that last time, but this time yeah. Okay.
Thank you very much.
Thank you so much.
Thank you.